In this video, I'll do a walkthrough of the different specimens for Lab 24, which are vectors and pathogens associated with circulatory and lymphatic diseases. So first up here, we have another flea. So this will be the third flea that we've looked at. This is Xenocilla, sometimes referred to as the oriental rat flea. But this is important and studied in this particular chapter because it can spread plague. Now, what I want you to notice that's going to help you distinguish this flea from the others is notice the parallel rows of hair on its back. This is different from the human flea and the cat flea that we've looked at. Also notice the leg, the back leg here, are going to have these really long hairs on them. And they're also going to have kind of this double hook structure on the ends of their legs, whereas before we just saw one of those. So this is Xenocilla, the oriental rat flea that spreads plague. The next one we're going to look at here, this is Pediculus humanus. This is the human body louse. So you see here it looks similar to the other lice that we've looked at. You do see all three of its legs kind of extended here, where that's a little different. We didn't see that with a couple of the others that we looked at before. Long oval body here, kind of the little horns as we've alluded to them before, ribbed. Okay, you can kind of see the segments in them. Notice the very long pinchers on the first set of claws here. Okay, we see them on all of them, but on the first set, really pronounced set of pinchers there. So Pediculus humanus. Now we're going to switch over and look at a couple of different ticks. So we've already looked at the Lone Star tick. Now we're going to look at a couple of others that are important in spreading disease. This first one that we have here is Dermacentor variabilis, sometimes referred to as the dog tick. But what I want you to notice is the carapace here. Remember, we talked about this with the Lone Star tick, this shield here on their back. And then notice the body here, no spots. Remember, the Lone Star tick had a little spot right here. We don't see that on Dermacentor. The next one we're going to look at is Ixodes, Ixodes scapularis. And this Ixodes is the primary vector for Lyme disease. And for Ixodes, you're going to look at the female and also the larval stage. Now, remember I told you that ticks are related to spiders because they're arachnids. They have 8 legs. What I want you to notice about the larval stage is that it only has 6 legs. The larval stage is sometimes referred to as seed ticks because they're much, much smaller. They're about the size of like a strawberry seed. And that's where they got the name seed ticks. But they can spread disease just like the adults, okay? Now, with the Ixodes scapularis, what you're going to notice is that it's a very large carapace. So this shield, this carapace on the back, is about half the size of the overall female tick here. And then her body is very light in color compared to the others we've seen. You'll also notice her legs are almost striped in appearance. Okay, so you've got kind of these little striped markings on the legs. So make sure you get the adult female here as well as the larval stage. And again, the key here looking at the larval stage is we only see 6 legs. Okay, then when it goes into the nymph and the adult, we get to 8 legs. We're going to take a look now at a couple of mosquitoes. So the first one we have here is Aedes aegypti, and this is the male. Okay, now Aedes mosquitoes are black and white. And so you can see the body here with these little white spots, okay, all down, and these would be on both sides of the body, okay. Now this is the male. And when I look at the male, how I'm going to see this is different than the female. I'm going to look at the antenna. So you see the antenna here. Notice how bushy the antenna are, okay. Very wispy, very bushy, okay. So this would be the male. The male is also going to have a skinnier abdomen. Okay, so you notice his abdomen is very skinny. So the male is going to have a very skinny abdomen. Remember that males don't take blood meals. Okay, only the females take the blood meals. So this would be a male Aedes aegypti. Aedes mosquitoes spread yellow fever, among others. Now, when I take a look at the female, okay, so here's the female. Notice the antenna. Okay, we still see the white spots along the abdomen. Notice the abdomen is thicker, and then the legs have very distinctive white markings on them, okay, particularly where they're jointed. So I'm going to go back. Here's the male, okay, white spots on the abdomen, a few little white spots on the legs, but not very many, and then very bushy antenna versus the female, very nondescript antenna, and very, very pronounced white spots on the legs. Okay, so that's Aedes aegypti. Then I'm going to skip the Plasmodium vivax for a second and go to Anopheles. So Anopheles is our vector for malaria. So this is Anopheles. Anopheles is a very dark brown, almost black color. And you'll notice here its wings kind of laid over on top of one another, kind of this glossy appearance on the leaves. And the one thing I want you to notice that's going to set the Anopheles apart is look at this stripe right here on its thorax. Okay, and sometimes we'll see more than one stripe on the thorax, but very, very pronounced stripe on the thorax. So we have Aedes that's black and white. We have Anopheles that's dark brown with this stripe on its thorax. And then with Lab 25, we're going to look at Culex. And you'll notice it's a light brown color compared to these other two. So that's Anopheles. Now I'm going to go back to Plasmodium vivax. So this is a blood smear of Plasmodium vivax, which is one of the species that can cause malaria. And what you're looking at here is a blood smear. So all of these are going to be red blood cells. And then right here in the middle, this is a stage in the life cycle referred to as a schizont. Okay. And what's going to happen is that these are immature forms, and this is going to rupture, and all of these little pieces that you see in here are going to filter out into the blood and begin to infect other red blood cells. Okay. You also see a little piece of pigment, this brown right here. What you're going to see when we talk about Plasmodium is that Plasmodium has a very extensive, complicated life cycle, and it takes on a lot of different forms within the blood and the liver. And this is what allows it to cause the cyclical symptoms that it does. And so this is just one form that we would see in the life cycle of Plasmodium. Now we're going to look at another protozoan here. This is Toxoplasma gondii, which causes toxoplasmosis. And again, this is a protozoan. They kind of have this crescent shape here, okay? Large, dark staining nucleus, kind of off center, okay? So that's Toxoplasma. Now we'll look at the assassin bug, or the triatomid bug. This is the causative agent of Trypanosoma cruzi. In the Lab 25 video, we'll see a blood smear of Trypanosoma brucei, which is very closely related to Trypanosoma cruzi. Trypanosoma cruzi causes Chagas disease, and its vector is this assassin bug or triatomid bug, or sometimes called a kissing bug because it likes to bite on the face. We are starting to see cases of Chagas disease in the United States. This triatomid bug is making its way north. It looks similar to what we call a stink bug, but it's larger. And it has this kind of darker triangular shaped structure here on its back. And then here, of course, you see its head here. So this is the triatomid bug, or assassin bug, kissing bug. That is the vector for Chagas disease. Now, our last few things that we're going to look at here are for a particular organism called Schistosoma mansoni, which is another type of fluke. We looked at Fasciola hepatica in Lab 22, and Schistosoma mansoni is also a fluke. Let me kind of come down here. And it looks a little bit different. There we go. Than the other type of fluke, and it has a very complicated life cycle. Schistosoma has a primary host in a particular type of snail. They're going to be in water. They're going to infect this snail and then have other free living stages that become infected. So I'm going to go a little out of order and walk through the stages of the life cycle kind of in order. So the first thing I'm going to look at are the eggs. And we're going to start with, I'm going to look at this one right here. This one is very characteristic. Oops, I didn't mean to do that. Pardon my fail there of closing out my windows. So this is Schistosoma egg. And you're going to notice this looks a lot different than the other eggs because it's got this little projectile kind of coming out here. It kind of reminds me of, like, if you're looking at comics and you have a little conversation bubble. That's what the Schistosoma egg reminds me of. Notice that we don't really see a thick wall. We have no empty space or no clearing. And we have this little part this little kind of pointed part that comes off of the egg. The second step in the life cycle, oops, not that one, is the Myristidia stage. And this would be a free swimming stage here. So this little piece in the middle that you're looking at is the Myristidia. And I'm going to point out a couple of things here. Notice the cilia. Okay, this is free swimming, so it uses these cilia to swim around in the water. And you'll see these two little spots here. These are eye spots. So this is the Myristidia stage. If I go back to the life cycle, you can see here that these eggs, or the eggs hatch and release Myristidia. The Myristidia then are going to infect the snail. And after they infect the snail, we're going to get the sporocyst form. And this is our sporocyst. So the sporocyst is basically an elongated form of the Miracidia without the cilia, because this is what's going to develop in the snail. We may see a few of these swimming around free, but primarily, as you see on the life cycle, the sporocysts develop in the snail. And there's going to be several generations of these. We have primary sporocysts, and then they'll change a little bit and become secondary, and so on. And then eventually these are going to basically become mature into this structure called the Circulatory. Let me find that one. There we go. So this would be what would hatch out of the snail, or what the snail would release. And these would also be free swimming. Notice they have a really unique shape. Their head region here kind of looks like the sporocyst, but then they develop this little tail. And this allows them to swim through water. And this is what would infect the human. And then it would develop into the male and female. And here we go. Here would be the male and female. Now, here's the other really odd thing about Schistosoma, is the male and female are interconnected. So this larger structure you see here is the male. The female actually resides partially within the male, as you can kind of see here, okay? And so then they would reproduce. They would produce the eggs, which would be shed in urine or feces. And then those would get into water, they would hatch, and we would repeat that life cycle over again. So let me go through these again, just because I know I kind of went fast through these. So first we have the egg. Okay, so this is the egg. And then the egg would hatch into the Miracidia, which kind of looks like the shape of the egg, but then has cilia and these eye spots around it. This is what would infect the snail. And then inside the snail, we would have the sporocysts. Notice they're non-ciliated. They're not free living anymore. They don't have a need to be able to swim around. And these would go through successive stages of development. And eventually out of the snail, we would get the Circaria. This is what would infect the humans. And then we would get to the adults here. And again, the male and the female here essentially live together. If you want to draw these in a single circle, you can, and just label them male and female. So that brings us to the end of our vectors and pathogens for Lab 24 diseases of the circulatory and lymphatic system.